Today, I would like to discuss with you our new finding of how the growth factor Wnt is a regulator of endocytosis. And this will illustrate how basic cell biology or curiosity-driven research can impact sometimes, perhaps, practical problems. The growth factor Wnt is of interest to us because it is important in early development. See? In the early embryo on the left side, you can see it's the earliest signal that the embryo has. In fact, it interests many people because the growth factor Wnt is very important in cancer as well as in stem cells. And Roel Nuse has worked out the canonical Wnt signaling. When Wnt is off, you see that beta-catenin, the yellow protein, is degraded very rapidly. It's phosphorylated by an enzyme called GSK3, which gets ubiquitinated and goes into the proteasome. But when Wnt is on the right-hand side, then beta-catenin is not degraded, and it goes into the nucleus, where it activates Wnt target genes. So our contribution to this field has been to, I would like to say that usually Wnt causes cancer, not because there is too much Wnt, but because you mutate these two proteins called axin or APC, adenopolyposis coli, that are necessary for beta-catenin to be degraded. So if you don't have those proteins, beta-catenin becomes stable. And our contribution to this field has been to realize that in addition, in order to get Wnt signaling, this enzyme GSK3 must be sequestered inside these multivesicular endosomes seen there at the middle of the screen. So then when the GSK3 together with the Wnt receptor complex is inside these vesicles, it becomes sequestered from the cytoplasm, and therefore beta-catenin, here in blue, is stabilized, goes to the nucleus, and causes all these changes. The multivesicular endosomes, or multivesicular bodies, go into the lysosome, where then they are degraded. And in this paper, some years ago, we found not only that Wnt stabilizes beta-catenin, as was known, but rather than up to 20% of human proteins have GSK3 sites as well, and they're stabilized by Wnt. So this is now like a whole field of research. And when you find then that what happens is when you activate Wnt signaling, then GSK3, which is pointed there by the arrows, moves into these multivesicular bodies, and which have all these vesicles in them. And so it moves from the cytoplasm to inside these membrane vesicles. And this might seem a very strange thing, but this is just part of the normal process of endocytosis in cells and their membrane trafficking. You will see it here in a little diagram. So you get endocytosis. And these here are going to form these intraluminal vesicles of the multivesicular bodies, and they go to the lysosome. And then you keep on taking out proteins from the outside, and then we're going to form again some intraluminal vesicles here, these multivesicular bodies, and these are then secreted to the outside. So what we have realized recently is that when we give a short treatment of Wnt to cells, they form these very large vesicles, which are multivesicular bodies. And you can see on the right that there are the very large vesicles after only 20 minutes of Wnt treatment, as opposed to the left where there are very little vesicles. And these form very rapidly within 5 minutes, and the result of endocytosis, because we can add a protein labeled with fluorescein called bovine serum albumin, and with Wnt, you see in the red curve, you have much more endocytosis. And when you take in the bottom part of the slide, we have another reagent called BSA dequenched. So this is a protein, you put it in the culture medium, it fluoresces only when it is degraded inside the lysosome. So this means that the protein is going from the outside to the inside of the cell, and it is being digested. And so you can see that a little bit better in a movie here, where you see these cells that are taking up the BSA DQ, and when that one is cleaved in the lysosome, then the amino acids will be consumed. So this implies that when a cell gets Wnt, it's feeding from the outside and implies important metabolic changes, we think. In fact, in the serum, we have 50 milligrams per ml of albumin and other proteins. So a cell that is in the presence of Wnt is now consuming proteins from the outside. And this is something called pinocytosis, and this is a non-receptor mediated phenomenon. And pinocytosis, there are many kinds. One of them is the macropinocytosis, which means taking in big drinks. And so this is a cell, and you're going to see on the left-hand side the vesicles, that when they're over 200 nanometers, these are about a whole micron, when they are drinking from the outside, and hopefully you can see some vesicles going to the inside of the cell. Let me see it again. Okay, well. To study macropinocytosis, we have reagents like one called the dextran of a certain radius of above 200 nanometers of diameter. And when we do that, and we now activate Wnt by changing these tumor suppressors, that is axin or APC, when we knock these down, you see that there is an increase in macropinocytosis of dextran from the outside. And so that means that normally the function of these proteins is to repress the uptake of proteins from the outside. And this APC is a very important gene, because there are people that have a heterozygous mutation, just one gene mutated, on the adenopolyposis coli, and they develop hundreds of benign tumors of the intestine once they lose the second copy of the gene, and almost invariably they end up with colon cancer. And so there are many colon cancer lines that actually, this is just lowering the amount of the APC, but there are many colon cancer lines that have no APC at all. One of them I'm going to show you now in a very brief movie, and you see that this, in the absence of APC, this cell line, SW40, takes up in half an hour a lot of BSA DQ, meaning it's feeding from the outside. But if we replace the APC gene, so now it's APC, the only difference between these two cells is just one gene. Now these cells are not feeding from the outside. So there's a very big change from these cells. They're now feeding from the outside in the presence of the lack of APC or the presence of Wnt, and from the inside. And so there are many ways of inhibiting endocytosis and macropinocytosis. So this suggests the vulnerable point for these cancer cells, which is that we can attack them through the endocytosis. And this is entirely new. And we have tried one of these inhibitors that for macropinocytosis, that it is known. Macropinocytosis can be specifically inhibited by derivatives of the commonly used diuretic amiloride, which is in human use. The analog we use is IPA. And this blocks a sodium proton exchanger that is needed for this macropinocytosis, that will be needed for formation of these vesicles and sequestering the GSK3. Now these cancer cells, they have beta-catenin in the nucleus because they are cancerous. But if you put this blocker of the sodium proton pump, now within 2 hours, the beta-catenin goes into the cytoplasm. So there is hope that this will help, maybe depending on the concentrations. And there are other methods of affecting endocytosis. So in conclusion, understanding basic cell biology of cell signaling can sometimes lead to useful, possibly useful things just out of curiosity. And with that, I'd like to thank my research group. This work was done by Nidia Tejeda and Lauren Albrecht, and I'd like to thank my faithful secretary, who follows me even to the Pontifical Academy of Sciences. Thank you.